So this is actually how I ended up on the dark side, working for a manufacturer. So my interest in how oxygen works, and particularly how oxygen conserving device works, is what got the attention of some manufacturers when I was just a lowly home respiratory therapist working at a little company in Mentor, Ohio. And so I've been doing pulse dose related, oxygen conserver related research or product development since the mid-1990s, mid to late 90s. And it's part of my passion, the few things I found that I was really good at or liked. I probably liked more than I was good at, but I'll let you be the judge on that. So these are the objectives. I won't waste any time on that. Same disclosures, I work for a medical device company, and I work for free for the American Respiratory Care Foundation. So, similar but not the same. So when we talk about home care and home oxygen, it's a little different than when we talk about what most of us were trained to do as acute care respiratory therapists, where we generally slap oxygen on a lot of people, whether or not there's really an absolute medical need for it. And if you ever trained as an EMT, and that's where I started many years ago as a Navy corpsman, an EMT, then an LPN, then an RT, we slapped oxygen on everybody that got in the ambulance, whether you broke your ankle or you had hemorrhoids. We were given oxygen. And for the most part, because generally speaking at low flows, it's a fairly benign therapy. There's more risk of good than there is risk of bad. And so we use it much more broadly and across a lot of different reasons, mostly to treat any acute illness where there's a risk of hypoxemia. So, and we're really, the goal when we're doing it in an acute care setting, whether it's EMS or emergency room or on the floor, is to deal with that hypoxemia and reverse that hypoxemia. We're looking to prevent hypoxia. And again, we're much more likely to treat what really isn't true hypoxemia in the acute care setting, even if it's very mild hypoxemia. And in COVID, we were treating people with 92, 94 SATs because they were getting silent hypoxemia. And now we've also learned that the pulse oximeters, particularly in darker toned and darker skin tones, have been a lot more inaccurate, especially the less sophisticated devices. It's always been a problem. It's been a known problem. All of you are respiratory therapists. You always know we had color and nail polish issues and stuff. I don't think we knew how bad the problem was. I mean, the FDA's taken a very serious interest in it because it was underestimating. It was underestimating the severity of the hypoxemia and overestimating oxygenation in darker skin tone individuals. So they may have been undertreated, and that's a serious concern. So, but yeah, so we started treating COVID a lot more aggressively. Even with Medicare, pulling back the requirements from the 88%, recognizing that 92, and a lot of hospitals had 92 or 94% thresholds to still send you home on oxygen, which is a pretty long walk from 88%, which is where you start to get the edge of the oxyhemoglobin dissociation curve. So, but when you compare that to long-term oxygen therapy, and this is where pulse dose devices or oxygen conserving devices tend to play a bigger role. It's a little different. I mean, we still want to oxygenate, we still want to reverse hypoxemia, but we're not trying to solve an immediate issue, and it's not a short-term therapy. We're looking at people that over the course of a lifetime from disease, whether it was from self-inflicted disease of smoking, which is probably still 90+% of COPD root cause or greater, or combinations of congestion, heart failure, and other things that cause chronic hypoxemia. We're looking at really just getting you back. It's not so much about over-oxygenating you or protecting from that minor desaturation. It's really just getting you back to what would be an age-appropriate baseline. So if your saturation was 90%, if you're 68 years old and you didn't damage your lungs, and your SAT 90, 91 would be good SAT, that's where we're trying to get you. We're not trying to make you 99 walking around, you're not a marathoner. And so the objectives are a little bit different, and it's often important to do that. So we're really talking about chronic disease where you have impaired gas exchange for physiologic reasons, whether it's parenchymal damage, from like an emphysema, or the combination of parenchymal and bronchial gas exchange issues. And so it's really more about long-term outcome. You do get short-term benefit. I mean, definitely if you take a chronically hypoxemic patient, when I started in the field, we didn't routinely treat people that were chronically hypoxemic. We sent you home in the 50-50 club. You probably remember those days. Your PO2 and your CO2 were right around 50. You went home. You were a little blue around the lips, a little blue around the fingertips, but you didn't die, at least not that day. You died prematurely, and that's what the research showed us. And that's how we ended up covering long-term oxygen therapy for people with chronic hypoxemia. And part of the reason we don't treat acute problems. That's why when you're just in for that chest pain, and once that's resolved, they're not going to send you home on oxygen or let you have it around just in case you get chest pain again, because that really isn't part of the treatment. We're not normalizing you, and that's really what we're doing. And it's really a life-sustaining concept as opposed to a life-saving or life-supporting. Oxygen by itself isn't life support. And I've had this argument with the FDA many times. I'm like, if you're dying and you're not breathing, do you want me to ventilate you or oxygenate you? Ventilate. I'd much rather ventilate you than oxygenate you. So most respiratory failures is hypercapnic, right? And even when it's hyperoxic, you still got to get the air in and out. So you still got to have a mechanism to move the air. So if I just throw a nasal cannula or a non-rebreather on you and you're not mechanically moving air, you're going to die with a really high PaO2. So I talked about these in my earlier talk, because they tie back to the reimbursement in home care, because it's from the foundation of these studies that Medicare and other payers recognize that treating chronic hypoxemia has clinical value and improves long-term morbidity and mortality. So the KNOT study, and I just want you to see the age of these studies. So the KNOT study was published in 1980 after years of enrollment. So the study was really a product of the Sugarloaf Conference in 1974 and didn't get published until 1980. I had hair when that got published. Same in 81. These are two similar studies, but not the same. And I'll talk about those just briefly. It's important to understand the foundation of this, because I think somewhere along the line, we forgot that there's a difference in long-term oxygen therapy than what we do in the hospital. And they are different therapies. And over the years, I've had nurses argue with me, and even some doctors, why do they have to PO2? I wrote the order, I want the oxygen. And I'm like, well, if you wrote an order for an Audi, and I took it to the dealership, would they give me one? Just because you have the power of the pen doesn't mean it's appropriate and medically necessary. So, the KNOT study. It's an interesting study, and in retrospect, I think if you talk to Dr. Petty and some of the other investigators in that study, they would have told you they would set it up differently if they could go back in time. But unfortunately, none of us get to do that. So it's very, on scale, in comparison, particularly with a lot of studies today that are multi-site, complex studies looking at a variety of different outcomes, the number of subjects is really small. 203 is a relatively small N for a study that's basically going to change the way we treat people for the rest of our careers. And there's been a lot of questions that have looked back at this, and I'll talk about a little of that in a second. But this is where this came from. So everybody who works in home care knows PaO2 of 55, less than 88%, It's like tattooed on you somewhere when you first go to work in a home care company, because that's how we get paid. You don't meet that, no money, no mission, right? So they didn't really do what I would, like I was there when NIH was asking for help. They didn't really have a control group. They had a semi-control group. So you either were put in the continuous group or the nocturnal only group. There was no treatment group, even though it was still standard of practice at that time not to routinely treat people who were stable and had a PaO2 of 55 or less than 55. And so, or the secondary criteria was this, which was a much smaller subset of patients. And that actually never got proved out that it made much of a difference in the less than 59 group. But if you look, there wasn't a world of difference between the two groups. So continuous wasn't always continuous. It was an average of just under 18 hours a day. So in the continuous group, there was at least 6 hours a day. the average person wasn't wearing their oxygen as part of that. And in the nocturnal group, they averaged 12 hours a day, which is a lot of sleep time, so that meant they wore it more than they were likely sleeping, you know, and you can see the standard deviations there. So what they did see was over the period of the study, which was intended to be 5 years, and it was cut short, that the mortality rate for the nocturnal group was almost 2 times that of the continuous group. And at that point in the study, I think it was about 3.5 years into the study, they felt it was unethical to continue to have a control group, that they demonstrated that there was a significant mortality difference in the group that wore it longer, and that study was stopped. And so the big conclusions were continuous O2 therapy reduces mortality, and if you talk to like Dr. Petty, and I had the opportunity to be with him a number of times over the years, some O2 is better than none, and more is better than nocturnal was kind of the concept. So, but I'll show you where the sum might not be as scientific as we thought when you look at the BMRC study. So it is the largest and most frequently studied reference study. I mean, I was fortunate to be invited to the 5th consensus conference on long-term oxygen therapy and the 6th consensus conference, which were, you know, all the thought leaders, the Dr. Pettys, the Neil McIntyres, and all these brilliant folks. And then a few of us scoundrels from the DME industry got invited. And every conference starts with the NOT study and pretty much ends with the NOT study, up until the LOT study, which came along just more recently. So this is the group, and here's the most interesting takeaway for me, especially the first time I heard this presented to me this way, was, and this is where I really appreciated the difference between acute treatment and chronic treatment. Look where survival became statistically significant. It wasn't after a week or a month or even a year. It was 18 months in the NOT study. So there was no difference in survival in either population of people until you got out past 1.5 years. So that's when I say oxygen isn't life supporting. Long-term oxygen therapy. I'm not talking about what you do in the emergency room when you're whacking on somebody's chest and pumping them, you know, that's a whole different scenario. I'm talking about a stable person who's got mild to moderate hypoxemia, who's going home, who's got a chronic condition that's not going to likely get better. What makes them survive longer and extends the quality of their life? And so you can see that it really wasn't until you started to get out, and then it started to really separate, and you can see there was a pretty obvious, you know, this is statistical, which is for the statisticians. That's just math. That doesn't mean it's clinically relevant. This is clinically relevant. Which group do you want to be in? Which group do you want your family member to be in, right? I'd much rather pick that group. So BMRC study, smaller N, but a real control group versus a pseudo control group. So you either got oxygen or you didn't. 7 patients, and they focused on an average of 15 hours a day. It was the target for being on oxygen, and then obviously no oxygen if you were in the other group. Over 5 years, they held on to their study. They did take it to the end. And they saw an improved survival in the treatment group versus the control group with 55% survival rate versus 33% survival rate over the course of a 5-year look. So this is the BMRC study, and interestingly, they didn't see any difference either until 500 days. So just about 1.5 years. Pretty similar to the, so in this study though, you either wore it for 15 hours a day or more, or you didn't get it at all. So their survival, there was no difference between nocturnal patients and no oxygen patients if you overlap these studies. And there's been I heard a lot of impressive pulmonologists argue over that and disagree with it or agree with it. We want to believe that some oxygen is better than no oxygen, but didn't always work out for us in the research. So this is the summary of these 2 principal landmark randomized controlled studies. Is basically if you have severe chronic hypoxemia. The other thing to remember, This wasn't putting them on a pulse ox for 2 seconds, whacking them around, making them fake their finger, it goes below 88%, and they qualified, because I have seen people do that. This was sitting in a chair resting off oxygen for 20 minutes. Then they drew a blood gas, and you had to have a PO2 of 55. So this was in my best. This was off antibiotics, off steroids also for 2 weeks, a 2-week washout period. So this was the healthiest I was going to be at my most chronic. And you can see in this population, if you were on, basically it was nasal cannula oxygen, average was 2 liters per minute. For 16 to 18 hours or greater, you're going to have improved survival over someone who didn't get that oxygen. And that's really, if you go and you look at the World Health Organization, if you go and look at the gold standards, if you go and look at the standards in the UK, if you look at the standards in South America, most of them basically say for treating chronic hypoxemia and lung disease, 16 hours a day of use is the threshold, and maintaining a saturation greater than 88%. And why does it work? Nobody really knows. I mean, I asked the Dr. Petties, and they said, that's a great question. We think it was a result of, hopefully stopping that bad behavior that was the primary contributor, which was generally smoking, being more health conscious, obviously other supportive drug therapies, but really some remodeling of the lung, remodeling of blood flow into the major organ systems, improving blood flow and oxygenation. That is really what caused, prolonged life expectancy in the population that survived longer. So, and that's why when they fought like doing retesting and then taking people off oxygen, there was a strong pushback from the pulmonary community because there is a therapeutic benefit. So if we put them on it and for 2 years or 3 years and we get this remodeling, and I've seen it with my own eyes, and you take them off oxygen for 6 hours and their SAT's above 90. So technically they don't qualify. You could take it away from them. But what happens when they get a cold? What happens as they continue to decompensate? So there's a therapeutic benefit. If I put you on blood pressure medication and your blood pressure goes down, do I take you off of it? And that was kind of the logic that was argued when we fought against it, because Medicare did want to retest everybody at 90 days. And then once a year. So this was just a study that later, and this is where it kind of ties into the importance of why there's been so much emphasis on ambulation, and particularly devices that encourage ambulation, like portable cylinders, transfilling devices, portable concentrators, is the more you walk, the better, the longer you live. And I would say this ties to, as I age, and that means everybody around me is getting older, parents, relatives, movement, being active is probably the greatest prevention tool, and I think that's what this study basically showed, that if you were on oxygen and you were in the low walk, in the low walk group, you still did better than the nocturnal or no oxygen, but just this is nocturnal. The high walkers who were in the continuous group obviously had the best outcome, and this is the survival curves. So you can see low walking on oxygen and high walking just nocturnal were actually pretty similar in the way their curves flattened out. And then the worst outcome was the low walkers who were only wearing their oxygen nocturnally. They had the worst outcome. You could argue they were probably the sickest. Maybe that's why they weren't walking, because it's hard to control for those kind of elements. But those are some interesting studies they're very old, but they're still relevant because there's nothing new. So sometimes being old is good. This is the most recent study. This was published in 2017 in the New England Journal of Medicine. So after all that time went by and we've argued and argued and argued over the NOT study and its relevance and the BMRC study and its relevance and whether or not it properly reflects who we need to treat and when we should treat, there was a strong argument from one side of the pulmonary world that said we should treat people earlier. The NOT study waited too long. We waited to them to get too sick. We waited them to get too chronic. We should intervene earlier. Maybe a PO2 of 60 is the right threshold at the top of the oxyhemoglobin dissociation curve, not in the steeper part of the fall. And so they proposed a study, multi-year study, through the NIH. And Medicare actually funded this indirectly by allowing coverage for those patients enrolled in this study, because they actually were believing that they was going to prove that less people were going to benefit from oxygen, and it was going to lower the risk of them paying for oxygen in particular. And so this study was founded. It ended up with 738 patients, 42 centers, ranged from 1 to 6 years in the study. They picked a population that wouldn't normally qualify for oxygen. So if your saturation was greater than 89%, and you had or an exertional during a 6-minute walk test where you were less than 90 but greater than 80. Those are the two thresholds for getting in. They didn't look at nocturnal, which is probably a good thing. Nobody wanted to screw with that here in the United States because we're just not sure. The BMRC study, infers that treating nocturnal only might not do much, but we have so much overlap between OSA, hypoventilation, and COPD, and just the normal hypoventilation that comes from the positional changes and the way physics works against COPD patients and their diaphragm movement and stuff, that nobody wanted to screw with that. So they didn't really look at the nocturnal only group. But there's, 30% of the patients who are prescribed home oxygen are prescribed nocturnal only, just based on an overnight oximetry study. So they looked at basically that group that didn't qualify at rest but qualified with exercise, or were in that what used to be the borderline group, that were like this PaO2 of 60 or a saturation 89, 90. And that's what they took a look at. So they were randomized. They were either in the O2 group or they weren't. They either got 2 liters a minute, 24 hours a day. The exertion group, if you qualified only during exertion, you'd used it with exertion and sleep. So they did address nocturnal. There is a correlation in some of the patients with nocturnal oxygen that if you do an overnight oximetry and they desaturate, they also desaturate during exercise. There's a couple studies out there. If you spend some time researching, you can find them. So they concluded that, and just so you see, the ambulatory dose was individually prescribed, but 2 liters was the average to keep a saturation above 90 for at least 2 minutes when they're walking. In the patients with stable COPD and resting or exercise-induced moderate desaturation, oxygen did not result in a longer time to death, first hospitalization, than no oxygen at all. Nor did it provide any sustained benefit with any regard to any other measured outcomes. And the measured outcomes, it's a really good study. I encourage you to read it. You can get most of these studies from New England Journal of Medicine. Like this kind of landmark type study, they're free. If you go to Google Scholar or any of the websites, Google Scholar usually gets you there. But this was not the outcome we all expected, I can tell you that, including talking to investigators that were part of it. And there's still been controversial. Some of them don't like the results and have said, well, it didn't change my behavior. I'm still going to prescribe oxygen the way I prescribe it. Others said, I'm not going to prescribe oxygen for exertion only. It doesn't do any good. And so it's gotten mixed reviews, but it is what it is. I mean, one of the risks when you do a study is the null hypothesis, right? You don't get the answer you want. You don't always get the answer you want. All right, pulse dosing. We're going to spend the rest of this talking about what I know and have learned and what's out there in the literature around pulse dosing. Does it work? Does it work well? How does it correlate to continuous flow? Some of and all the other things around it. And despite being around a long, long time, there's still a lot of, in Europe, for example, they don't use pulse dosing nearly to the level we do. I would argue here, if you're ambulatory stable oxygen user, and Kim, you're in the home care business, everybody's got a pulse dose, right? Something, whether it's part of a portable oxygen device, a concentrator, whether it's on a small cylinder, whether it's even a small liquid, they're all pulse dosing. So why? Well, if you took a team of brilliant engineers and said, let's make a really efficient, accurate oxygen delivery system, you wouldn't make a nasal cannula. Because that ain't what it was designed for. It's not accurate. It's not about giving, accurate FiO2. In fact, the FiO2 moves every time your breathing pattern changes. It's about supplementing, and then you make that titration based on your blood gas or your saturation or what other input the doctor wants, but generally PaO2 or SpO2. Get it up, it's working. And that's really, it's a low flow system. All the low flow systems are generally inaccurate because by design, they are influenced by all the key variables: your tidal volume, your anatomic dead space, your respiratory rate, your IE ratio, and your inspiratory flows. So when you're breathing from a nasal cannula, and let's say your average inspiratory flow is 30 to 40 liters per minute, and you're getting 2 liters per minute, where's the rest of the air coming from? Not coming from the nasal cannula. So it's a very, very, very highly diluted amount of oxygen by the time it gets here and to here. And that's why they're not efficient. That's why, but they work, because most of the people we're treating in long-term or chronic care are on the edge. And when you give them 2 liters, and why 2? I mean, Dr. Petty said if they went back in time, he wouldn't have just put so many people on 2. They would have individually really focused on individual titration. But they didn't have pulse oximeters back then. They didn't have the ability, and they had to pick a number that was oxygenating the largest population without an adjustment. And 2 over-oxygenated some, and was just barely enough for others. And then for the few that fell out, then they did adjust those patients to 3 or 4, but the average was 2. So I always said, why does everybody get prescribed 2? Why not 1? Why not 3/4? Why not a half? For the same reason we prescribe 2 Tylenol. Now, if you're 400 pounds or 40 pounds, 2 Tylenol. If you're 6'8", giant football player, I guarantee 2 Tylenol ain't touching it. But that's what we do, because, be honest, we learn to prescribe like we learn a lot of other tasks. And I was in the pharmacy business for a long time. Omnicare used to keep track of all this stuff. They were really tight with Philadelphia College of Pharmacy and stuff. Do you know what the average physician, how many prescriptions they can prescribe off the top of their head accurately? 5 to 6. 5 to 6 drugs. Do you know how many articles of literature a day a physician would need to read to be current with all of the prescribing practices of all the drugs that were on the market at any given time? They'd have to read anywhere from like 4 to 10 journal articles a day. Where do they get most of their education? The drug rep and the sampler. So anyhow, so 2 cured everything from hemorrhoids to brain tumors, and that's why we landed on 2. And that's why those of you who do home care have walked into a patient's home and found them 99% on 2, and have found them at 88% on 2. Because it doesn't work for everybody, and that's the the way we do it. So, but 2 liters is the majority. So simple math here and simple relationship. When you have a continuous flow, you're wasting a lot of gas in between I times and E times, and this is your window. The area under the curve here is your opportunity to get oxygen into the lungs. So the interaction between flow, inspiratory flow, and the cannula determines how much oxygen gets into the airway. So I used to use this when I taught patients about oxygen when I'd go to the pulmonary rehab groups and stuff when I was back in home care. There is all flow is volume. That's why it's liters per minute, cc's per minute. It's a volume over time. So you don't fill a bathtub with flow. You end up with volume when the water comes out, right? So basically, your liter per minute converts to milliliters per second, and these are your influencers. And like I always say, if you're on 2 liters per minute, and if I put all of you on 2 liters per minute, what would be the amount of oxygen that actually made it into each of your lungs? It would be different for every one of you. Because if I asked you all to take a sip of water, would you all drink exactly the same amount of water? That's how a nasal cannula works. Done. We now know nasal cannulas aren't great. But they work really, really well for the intended purpose. So, what about purity? When concentrators first came out, there was a lot of pushback. Well, they don't make 99.996 oxygen. Yeah, so what? Do you need 99.996 oxygen when you're getting most of the oxygen from the air anyhow? So it's a dilution ratio, right? So this is actually from any textbook on oxygen. You'll have a nasal cannula FiO2 equation. I don't know what color your Egan's or Shapiro's was. That's how we used to gauge when you became a respiratory therapist. I don't know if they do that anymore, but You have the green Shapiros, the yellow Shapiros, or the red Shapiros, the green Egans or the blue Egans. But anyway, if you can find those old books and you dusted them off, or if you find a new book on home respiratory equipment or respiratory technology or something, you'll still find these equations. But this is really how little influence the purity of the gas has within reason, because so much of the gas that you inhale comes from the room, not from the source gas. So the difference between 100% and 85%, which is the bottom threshold of a concentrator, on a nasal cannula at 2 liters per minute, and these are the factors I've used in the assumption, typical tidal volume, I time, is not measurable by any oxygen analyzer you have unless you have a Servomex. And I guarantee you're not doing home visits with a Servomex, because it's like a $7,000 oxygen analyzer. So you couldn't even measure the difference accurately with an analyzer. So when someone says to you, concentrators don't give you as good oxygen, now you can tell them they're full of crap. At least when it applies to nasal cannula. Now I want to preface this by saying this only applies to nasal cannulas and only nasal cannula low flow. So we're not talking about high flow nasal cannula. That's a whole different animal. The stuff that you guys were throwing on COVID patients all over the place. Not to knock it, but there was a nice study that showed that there was no difference in outcome between traditional high flow oxygen systems and high flow nasal cannulas in COVID patients that were hypoxemic, but that's for another lecture. So fundamentally, when do these things start? Well, the early ones were in the early 80s. Puritan Bennett had a patent that went back, I think, 1983, which was a CR50, and it was incorporated actually in a liquid vessel. And then they, to make the liquid last longer, and then it was subsequently put as a standalone device to make cylinders last longer. And it was a very simple device, very simple demand valve. Worked like, those of you who remember the Bennett PR2s, the Bennett valves, and or Birdmark 7s, Birdmark 8s. I'm way too old for most of this audience, but some of you remember those things. So it was a simple demand valve. You trigger negative pressure, open the valve, flow came out. As soon as the back pressure started to overcome the pressure of the valve, it closed the valve when you started to exhale. Operate very similar if you've ever worn a diving regulator. So when people say, well, can pulse dose really deliver an adequate amount? Can you breathe underwater with a regulator? Yeah. You don't breathe when you're exhaling, right? Remember that. I told you didn't have to be real smart to have my job. If I can do this stuff, trust me. So the first ones were basically built off the Bennett PR2 valve, real simple demand valve. They were actually a dual lumen. One side was just the sensing side, and the other side was the delivery side. And really came along in the late 90s. Precision Medical was the first company to develop and launch a single lumen where it sensed and delivered in the same valving system. It took them a while to get it right, but they did, and that's kind of the standard now of the pneumatic conservers is a single lumen. These were actually not built for POCs. They were built for liquid vessels and cylinders to make them last longer. And actually there was a whole period of time where they were looking at these in hospitals, and there was a couple of, Puritan Bennett had one, I know there was another company. They were looking at them at the bedside to save oxygen to the hospitals because patients leave the room, they leave the flows going, you know. And so similar to how you remember the ambu bags, we used to have that lever that you'd hang the bag over so it would shut the valve off so it wasn't running flush all the time. Same principle for the nasal cannulas when the patient was at x-ray for 6 hours and all that, and the tube, and we're oxygenating the hospital. So from a clinical perspective, we wanted people to move, right? One of the cool things about getting to know Dr. Petty was his passion about patients. He was up in Colorado. He was a National Jewish. So if you're a COPD patient and you want to pick the worst place to live, go up on a mountain. And so they have a disproportionate number of hypoxemic COPD patients because of altitude. Higher you go, thinner the air, you know, less partial pressure, less molar volume of oxygen, right? So he was very passionate about, and really what Dr. Petty used to say was, the whole purpose of this wasn't about Medicare and money and DME. It was about giving people the opportunity to live the same life and do the same activities or as close to those they had before oxygen after they were prescribed oxygen. That was the whole purpose. And he had a vision, and I was at the 5th Consensus Conference I think that was in like 1996 or something like that. I can't remember exactly what year it was. And he wanted a 5-pound, 2 liter per minute portable oxygen concentrator. That was what he thought was the panacea. If we could do that, then we really got a portable oxygen system. He called E cylinders and D cylinders schleppable. And I guess that's a Yiddish term, but draggable, you know, so he didn't like them. And so obviously, as we introduced more conservers, the use of lightweight, you know, when I first got in home care, you never saw an M6 or an M9. You got an E or an M at the house. Not an M9 or an M6 or one of these very small cylinders, because they wouldn't last for 15 minutes, you know. Now we have hours. And they're on everything. So this is it's an old photo. It gives you an idea how many were on the market between like the late 90s or mid 90s to early 2000s. Some of those are no longer on the market. Some of these are on the market. Every POC, every transfill device is tied to using a conserving device. That's a big principal part of the philosophy of them. These, by the way, are oxygen conserving devices. They were invented by Dr. Brian Type. Brian also invented the first electronic conserving device, and he licensed that to Chad Therapeutics. So some of you remember Chad Therapeutics. So the Oxyvisor, the mustache cannula, those were in Shapiro's, I think, even in the yellow one, because that's how old they were. Those patents date back to the 1980s. We still make them. Drive Medical bought the company that bought Chad's conservers. So it was a company called Innovo. Drive bought them. So they actually sell those. We still sell those today. And during COVID, we couldn't make them fast enough, particularly for Europe where they don't like conservers. So we've been doing this for like 30 years now. You know, conservers were introduced in the 80s. They became very popular in the 1990s when the Balanced Budget Act hit. We were all trying to reduce the number of deliveries we were doing, and we were reaching and grabbing. That's where my interest came in, because we'd never used a conserver before 1997 at my company, because we were getting paid enough that nobody really was trying to reduce the number of cylinders. We didn't care too much about that. We got a 30% cut, and things were starting to hurt. People were laying people off. And there were only a couple of conservers on the market at that time. And I remember that med trade that year. Purett and Bennett had had the CR50 on the market since the mid-1980s and had never sold out. They sold out the first day of med trade. They were on back order for almost the remainder of that year, because everybody wanted to try these conservers to see if they could reduce the number of tanks they had to deliver, because delivery was where all the cost was. So that became the thing. That's when I got an interest in them. And my first question was, well, do they work? And all the sales reps said, yep, they save oxygen. And I said, but clinically, do they work? And there were no studies. There were very few things. And that's how I got involved, and Bob McCoy and Pete Bliss and other people that you may have read papers from. But so are they perfect? Is 2 + 2 always 2? 2 = 2? Well, not always. Why? Because nasal cannulas aren't perfect. So the predicate is an inaccurate device. So how do you take an inaccurate device and say we're going to be as accurate as an inaccurate device? Well, you have to pick a number. So no matter what number you pick, it's going to be like a broken watch. It's going to be right sometimes, and it's going to be wrong sometimes. You just want to be right more often. And so here's the problem. It doesn't provide a constant and consistent oxygen delivery because of all the variables we talked about. So these are intended, but everybody has to make a decision when you're making and inventing a conserver to what assumptions you're going to put into that model to decide how big a bolus you're going to give, how you're going to deliver that bolus, and why. And most of the assumptions started in the same place, and then people went in different directions for different theoretical reasons or practical reasons. So they're not exactly the same. They're mathematical derivations of what continuous flow is doing under normal breathing circumstances. And I'll explain that in a second. You got to try to account for the patient's inspiratory flow, their anatomic reservoir, and the mechanical dead space and the anatomical dead space. The pulse dose, has revealed some things. when I first started working with pulse dose, we started asking the question, well, can you do it at night? We know, we did enough clinical work in my own business to know that most of the patients could tolerate one of the brands of conserva that were on the market at the time we started our work. There was electronic ones, there were pneumatic ones. We used a couple different ones. And then there were patients that didn't tolerate them at all, primarily pulmonary hypertension and pulmonary fibrotic patients. I'll give you my own theory on that in a second. But for the most part, your 2-liter, 3-liter COPD patient, otherwise stable, like 100% of them we would have no trouble with finding, as long as we titrated them and we found the right device that matched that individual patient. Kim's done is put as many people on pulse dosing devices as I have. It's about matching the product to the patient and getting the right product for the right patient, tied to their clinical needs and their lifestyle needs, because not everybody does the same thing. But it also, when we started looking at nocturnal use, everybody said, well, I would never put anybody on a pulse dose device at night. And I would say, well, why? Well, what if the cannula falls out? And I said, well, that's a brilliant question. What if the cannula falls out when they're on continuous flow? Defeat objection number 1. Well, what if they have hypopnea? What if they have a hypopnea? Well, they're not breathing. So it doesn't matter if they have continuous flow or no flow, other than arguing there's a reservoir, right? There's a reservoir flow. But if the device responds quickly when they do take that next breath, maybe you can overcome that reservoir effect. So, okay, that's your 2 for 2. What else you got for me? Shallow breathing. And I said, okay, you got me on that one. Sometimes the device won't trigger, but how much oxygen do you get when you're shallow breathing? Even if you have continuous flow, you still got to take it. It's still a water fountain. So just because it's there doesn't mean it's getting into the deep areas of the lung. So I called BS on them. And then I did a study, and we showed that most of the patients pulse dosed at night equal to or better than they did on continuous flow. We had one patient that didn't, and only because she kept taking it off all night because the sound bothered her. So we started, making sure the patients understood how to use it, and we let people use them at night. Because do COPD patients only sleep at night? If you've ever done home deliveries, you know they're asleep most of the day. In that recliner, in the living room, next to the ashtray. So, while Rob Chatburn, if you don't know who he is, he's one of our leading scientists in respiratory therapy. I have the good fortune, he's in Cleveland. He's a genius, and when I mean genius, I mean Goodwill Hunting genius. I've learned more from him about research than I ever learned in any college I've ever gone to or anything. He's absolutely brilliant. The first thing he told me when I started doing research under him was, most of the things you were taught are going to be wrong. It just may take time for us to figure that out. So if you look at our career, IPPB, chest PT, mucomist, I mean, the list goes on and on. High tidal volume, low PEEP. That's what we were taught at the Shapiro School, right? All wrong, right? So don't think you're right because this is the way we've done it. Science is the equalizer, right? Good research, not today's research, but good research. So what happened? Well, not everybody bought into it. We got great acceptance on daytime use, not so much in the rest of the world. Changing clinician beliefs and behaviors is hard stuff, man. The research on research of changing clinician behavior is, what's the cycle of time it takes from the time evidence is introduced until there's full adoption of that evidence? Take a guess. 16 years. 16 years. So if you want to think about that, think about the asthma guidelines. How long have the asthma guidelines been out, Kim? Since early 90s, right? When did they get full adoption? Never. There were still people ordering Theodore sprinkles for asthmatics in the early 2000s as a primary treatment for asthma. Because I call it, I haven't killed anybody yet theory. Been doing this way for 25 years. I haven't killed anybody yet. There are various markets. I would say today, here in North America, Canada and US in particular, ambulatory oxygen users are disproportionately using a pulse dose, with the exception of that subset of patients who just don't tolerate them. And we all know, you have a pulmonary hypertension, pulmonary fibrosis, some of these patients with other odd interstitial lung diseases and certain things. And my theory on that is most people have some pulmonary reserve. Most COPD, most patients with chronic lung conditions have some limited pulmonary reserve, not nearly the pulmonary reserve they had when they were healthy. And their minute volumes are relatively normal. When you sit next to a patient with severe pulmonary hypertension, their minute volume is not normal. And any bit of oxygen that's available, whether it's in the anatomic reservoir or the dead space, whatever they can get, they need. And for that group, when you take away that continuous flow, that little bit of flow that's there at the onset of inhalation, which is the millisecond delay that takes a conserving device to react, is enough to cause desaturation in that population. And that was my own personal experience. And so sometimes they're very difficult to oxygenate. Sometimes you can get them, sometimes you can't. So basic mechanics, this isn't rocket science at all. When I'm talking about the anatomic reservoir or the reservoir effect, it's this flow that's sitting in your mouth, your nose, and in the tubing of the flowing gas force that while you're exhaling and pausing. So for those of you who grew up in the 60s and 70s, the shotgunning effect of how a bong works, I have no experience with that. Gene gave me shady after he learned a little bit about my youth. So anyhow, I believe that plays a big role in why there are some patients who just aren't going to, the best conserver is not going to work on the absolute worst patient at times, but for the majority of patients in the subsets it does. So here's the math, right? So we all learned that the last third of your inspiration or inhalation is anatomic dead space, right? It doesn't mix. It's not a mixed gas. It doesn't make it into the lungs. It's the first part exhaled. That's why non-rebreathers masks work, right? You're exhaling the last part and it's unchanged. It's not a mixed gas. So that don't count. So you have this window, this area under the curve. That's where it shrinks. So that continuous flow, everything happening from here forward and in some patients from here backwards doesn't count. So all you have is that. And that's the fundamental concept that we start with when we're inventing a conserving device. And then you say, okay, how do I do it well and shut it off and save oxygen? Well, you just change the direction of that curve, right? So it's the same volume, you just do it faster. So the average pulse dose device delivers at flows ranging from about 6 to 15 or 18 liters per minute, depending on the device and the setting. So it's a very rapid bolus of oxygen, and it's delivering that bolus over time. So you're just speeding up the time, and you're delivering it. So you're delivering that volume over a shorter time, is all you're doing. You're just changing the metrics a little bit. So this is the breakdown. This is the fundamental math you start with when you're creating a conserving device. What everybody's done, because I worked with, before I got on the manufacturing side, when I wrote my first few papers and manufacturers came up to me and said, hey, we'd like your input. And I worked with Precision, I worked with DeVilbiss, I worked with, who else did I work with? Inogen in the beginning. They were interested in what we discovered in our investigation. So that's kind of where everybody starts, and then you start making adjustments based on your device and what you think your device does better than the other guy's device or whatever. And that's when you get into marketing and other crap. All right, so if you start with a regular nasal cannula, here's your fundamental assumptions. If you go normal IE ratio, 20 breaths per minute, which is actually much higher. If you do lit searches now, that came from the KNOT study, and those are really end-stage COPD patients. Our data shows, and my research has shown, that 16 to 18 is a much more common resting respiratory rate among COPD patients. I don't know what you guys see when you see them in the field. I mean, if you see them in acute care, it's probably a different number, but when you see them stable in the outpatient setting, that they're at rest, their resting respiratory rates are lower than the 20 that was always the hypothesis. But we use that because that's kind of the one standard deviation off the average. And so if you take away the 33% that doesn't interact in gas exchange and just sits in the plumbing, that's how much oxygen you get in a typical, making those breathing assumptions, right? 12 cc's at setting 1, 22, 33, 44, pretty straightforward. Again, not rocket science. So the first conservers that came out were 50-50. They were on during inspiration, off as soon as exhalation. They had some delay, so they weren't giving the whole time, but most of them were aiming at delivering somewhere between 12 and 16 milliliters per bolus per setting. The electric ones were doing it better at the higher end of the 16. Some of the early electronic ones were going very, very low as 8 cc's, believing that they were fast enough to make a difference. But basically, this is how it works. So in continuous flow, your breath rate and FiO2 are inversely related. The faster you breathe against the same continuous flow, the lower the FiO2 per breath you get. Do you disagree with me? I'll show you the math. Pulse dose volumes can be adjusted, so you can. So fixed bolus devices, or devices that give boluses not necessarily fixed, but bigger than you would get on your continuous flow, even if they're minute volume adjusted, can deliver more oxygen to you as your respiratory rate goes up than you would be getting if you stayed at that same device and your respiratory rate went up at 2 liters per minute. And that's really the bets we're making on a conserving device. So again, your respiratory rate shortens, the area under the curve changes, and the window of time you have to get in that curve, which means you have to increase your inspiratory flow rate here in order to keep up with the shorter, faster breath that the patient's taking as their respiratory rate goes up. And that's really what you have to think about. This is just a spreadsheet from some of my early work, but if you look at different IE ratios and you look at different breathing rates and you tidal volume adjust, you can see the range of bolus volumes you're going to get at different breathing rates for the same settings, because we're moving different gases along the same, different I times that are determining that. And this is from bench testing in a lab of continuous flow FiO2 measurements based on those breathing rates. So as you can see, and it's most pronounced when you're up here at 5, so at 15 breaths per minute, at 5 liters per minute, your FiO2 is 37, 38. And it's barely 30 when the respiratory rate goes up to 30 breaths per minute. So you can see there, it's rate dependent. FiO2 is rate dependent. But if I pick the same volume of oxygen here and deliver it here, my FiO2 will be the same or higher, because it'll take up a greater capacity of the inhalation. It's a simple dilution. So if I give more oxygen when they're breathing a shorter tidal volume, the FiO2 per breath goes up. But that's not all. There's other variables that make a conserving device work better than maybe another conserving device. Part of that is its sensitivity. So don't tell anybody outside of the room, because the FDA, but they really operate more like a ventilator than they do an oxygen device. They trigger, they sense, they deliver. Sounds like a ventilator, right? They're actually classified, the FDA classification of these devices is like a ventilator. I get a couple extra minutes, don't I? Because we had the whole CEU confusion at the onset. All right, so these are the variables that you have to contend with, right? Your bolus size, your sensitivity. This is the range of advertised sensitivities of the devices on the market today. So there's it's a pretty big difference between a 0.6 trigger and a 0.1. Here's my suggestion when I tell the engineers the trigger sensitivity I want. The most sensitive we can get where it doesn't auto cycle. And that's where you want to be. If you shake the tubing and it triggers, that's okay. Because I'd rather have it overly sensitive than undersensitive. The little extra oxygen is not going to hurt anybody if you get a bolus out of sync. If you can't trigger 5 breaths in a row, you're going to desaturate. And I don't want that. And so you can see, these aren't, again, it's not rocket science here. It's pretty basic physics. And the method, whether you do a fixed minute volume or a fixed bolus volume, all that really means is with a fixed bolus volume, you're predetermining the tidal volume every time. So I'm going to set it at 15 milliliters, and whether you're breathing 20 breaths per minute or 30 breaths per minute, it delivers 15. Or I base it on a maximum minute volume, and so like normal breathing against continuous flow, as you breathe shorter I times, there's less time for the device to fill the chamber behind it, so the bolus shrinks a little bit as your respiratory rate goes up. But generally speaking, it stays higher than what you would get on continuous flow. That's not true for every device, so I can't speak for every device. So, this is what happens when you get an inefficient design. You waste a lot of gas. You don't get nearly as much in. And that device might not be a bad device. It might be the wrong device for that patient who's more sensitive, needs a higher volume at that same setting. Maybe if I adjust the setting, that would correct it. I might have to put this one on 4 instead of 2 to get the same amount of saturation on that patient that they got on their continuous flow. This is from a study that Chatburn and one of his students did. I gave him a hypothesis. I said, we base all this on, we say, okay, patients with COPD have a diseased lung, but we don't count their breathing pattern as diseased, meaning if you have a prolonged exhalation, your anatomic reservoir shrinks or is gone. If I spend most of my time exhaling before my next breath, there is no anatomic reservoir. All of those equations were originally based on a 50 cc anatomic reservoir. So all those theories about how a nasal cannula, you've heard, 24 to 40% or 26 to 45% or whatever, it's all based on counting an extra 50 cc's in every breath. So he created a disease lung model versus a normal lung model with the same flows, and you can see there's a pretty significant difference in the FiO2 between those flow rates in a diseased lung that has a prolonged exhalation versus a healthy lung that doesn't have a prolonged exhalation, has a normal pause which allows the reservoir to fill, the mouth, the nose, the tubing. So a lot of COPD patients aren't getting nearly the FiO2 you hoped on continuous flow, and that's why sometimes matching 2 to 2 doesn't work and you have to set them on 3. I'm going to make it simple. No. Not always. Every conserver at some breathing rate is probably equal to continuous flow. It is like a broken watch, right? You're picking a number that's going to be wrong at some point in time for some breathing pattern under some condition, and you just want to try to get as much coverage, and that's what each manufacturer works on. And again, everybody makes different scientific assumptions, but what we do know is, and this is the best way I describe it, all conservers conserve. That's the easy part. Are all conservers good oxygenators? Well, I won't say there's bad in the sense that if you hit the right setting, you can probably get to where you want to be as far as the patient's oxygenation, but you may be running out of runway if you're on one that you have to get to setting 6 before you're equaling that patient on setting 2 on their continuous flow. They've made a lot of assumptions where they're making very low bolus sizes. Sometimes they do that so it says the tank will last 12 times longer. Well, I can make a tank last really long if I just shut it off. All right. I always say math is not an opinion. This is how it works, guys. Your boluses, you want more FiO2? You change the dilution ratio. How do you change the dilution ratio? Change the channel. Increase the bolus size. That's it. Simple math. And this is really the magic. Match the patient to the product. We don't do that as much, and it's harder to do these days when you're in business because you're trying to narrow your buying down, so you want to limit buying from multiple vendors. Hospitals do that notoriously, right? So you got to pick one device that's going to cover everybody, and sometimes you need more than one tool in that toolbox because COPD is not a homogeneous diagnosis, right? It's very heterogeneous, right? Just because 2 people have COPD doesn't mean they have the same needs and they breathe the same. Every study I've read says titrate the patient to their device. The AARC Clinical Practice Guideline on Long-Term Oxygen says titrate the device. I don't believe in the 6-minute walk test for home use. It wasn't intended as a titration study. It was intended as a measure of your physical capacity. It was designed to measure conditioning in the military. I don't think it's a great test for titrating a patient. I think the ADLs they do, if they have to walk up a flight of stairs, then that's their biggest challenge of the day. If they have to walk 50 yards down the driveway to the mailbox, that's their biggest challenge of the day. And titrate, most of the targets are greater than 90%. And I'm running out of time, so I want to apologize. I'm not going to give you all the references. You can do them. I just partial list. There's more stuff on how effective pulse dosing is than there has ever been a study that oxygen is good for you, including night. And not my own work. Lewarski, a well-known Polish researcher. Peter Gay, a well-known guy here in the US. standards for the diagnosis in ATS. These are other studies that were done looking at on-demand oxygen. This most recent one was done in a hospital. The resurgence of pulse dosing devices in hospitals again. This is a 2022 study. They're looking at, no difference in the patients who got pulse dosed oxygen who got continuous flow on low flows in a hospital setting. So it's making that's the regulator that can do both that was in the study. So if you're really interested, go read that study. It was pretty cool. So right now it's in every ambulatory device today. Every portable concentrator's got pulse dosing. That's the big secret behind them, right? Is they want to encourage ambulation among those patients because we know ambulation increases the likelihood of morbidity, improvements in morbidity and mortality. Everything's not the same. Know your device. Understand how it works. Try it on some patients. Don't give up if it doesn't work on one patient because it might work on the next 9. Again, there are a variety. I don't say one's bad and one is good. I say they're different. And you have to match your device. I always say be smarter than the tool you're working with. Otherwise, we don't need respiratory therapists and other clinicians. The machines can do machine learning now and AI, right? So you got to be smarter than the equipment you're working with. And there's great evidence. Do the research on your own if you don't believe me. And I'm happy to send you any of my studies. If there's anything, that's my personal email, not my work email. So if you're ever bored and you want one of those studies and you really want to read it, send me an email. I'll send you a study. I got my 2 minutes back. Thank you.